What is cellular respiration? Cellular respiration is a process by which your cells break down macromolecules like glucose to produce ATP. There are three stages in cellular respiration. The first stage is glycolysis, which is the partial breakdown of glucose to pyruvic acid. The second stage is the Krebs cycle, or the complete oxidation of pyruvate to release carbon dioxide. And finally, the electron transport chain, or the oxidation of NADH and FADH2 to generate ATP. Let's move on in the first stage of cellular respiration, which is glycolysis. In the stage 1 of glycolysis, a phosphate group is added to glucose in the cell cytoplasm by the action of enzyme hexokinase. In this, a phosphate group is transferred from ATP to glucose, forming glucose 6-phosphate. Next, in the second stage of glycolysis, the glucose 6-phosphate is isomerized into fructose 6-phosphate by the enzyme called phosphoglucometase. For the third step, the other ATP molecule transfers a phosphate group to fructose 6-phosphate and converts it into fructose 1,6-biphosphate by the action of the enzyme called phosphofructokinase. For the fourth step of glycolysis, the enzyme aldolase converts fructose 1,6-biphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, which are isomers of each other. For the step 5, triose phosphate isomerase converts dihydroxyacetone phosphate into glyceraldehyde 3-phosphate, which is the substrate in the successive step of glycolysis. For the sixth step of glycolysis, this step undergoes two reactions. For the first reaction, the enzyme glyceraldehyde 3-phosphate dehydrogenase transfers one hydrogen molecule from glyceraldehyde phosphate to nicotinamide adenine dinucleotide to form NADH2. For the second reaction, glyceraldehyde 3-phosphate dehydrogenase adds to a phosphate to the oxidized glyceraldehyde phosphate to form 1,3-biphosphoglycerate. For the seventh step, phosphate is transferred from 1,3-biphosphoglycerate to ADP to form ATP with the help of phosphoglycerokinase. Thus, two molecules of phosphoglycerate and ATP are obtained at the end of this reaction. Now for the eighth step, the phosphate of both the phosphoglycerate molecules is relocated from the third to the second carbon to yield two molecules of 2-phosphoglycerate by the enzyme phosphoglyceromutase. Now for the step 9 of glycolysis, the enzyme enolase removes a water molecule from 2-phosphoglycerate to form phosphoenol pyruvate. Now for the final step of glycolysis, a phosphate from phosphoenol pyruvate is transferred to ADP to form pyruvate and ATP by the action of pyruvate kinase. Two molecules of pyruvate and ATP are obtained as the end products. Now let's move on the second stage of cellular respiration, which is the Krebs cycle. Now for the first step of Krebs cycle, acetyl-CoA joins with oxaloacetate to form citrate. For the second step, citrate is converted to isocitrate. For the step 3 of Krebs cycle, isocitrate is oxidized to alpha-ketoglutarate, which results in the release of carbon dioxide. In this reaction, one NADH molecule is formed. The enzyme responsible for catalyzing this step is isocitrate dehydrogenase. This is a rate-limiting step as isocitrate dehydrogenase is an allosterically controlled enzyme. For the step 4, alpha-ketoglutarate is oxidized to form a 4-carbon molecule. This binds to coenzyme A, forming a succinyl-CoA. A second molecule of NADH is also produced alongside the second molecule of carbon dioxide. For step 5, succinyl-CoA is then converted to succinate, and one GTP molecule is produced. For step 6 of Krebs cycle, succinate is converted into fumarate and a molecule of FADH2 is produced. For step 7, fumarate is converted to malate, which is another 4-carbon molecule. For the last step of Krebs cycle, Malate is then converted into oxaloacetate, and a third molecule of NADH is also produced. Now let's move on the last stage of cellular respiration, which is the electron transport chain. The electron transport chain can be divided into four key processes, which are: first, the transfer of electrons by NADH and FADH2 to complex 1 and 2 respectively. Second, the establishment of an electrochemical gradient by proton pumping and movement of electrons through the chain. Third, splitting of oxygen molecule to form matter. And lastly, generation of ATP molecules by ATP synthase. In summary, glycolysis is a series of reactions that takes place in the cytosol. Glucose is a high in energy molecule, and glycolysis starts the process of extracting the energy from a glucose molecule. In glycolysis, it requires a glucose and 2 ATP molecules that will result into 4 ATP, 2 NADH, and 2 pyruvate molecules. So the net production of glycolysis is 2 ATP and 2 NADH that will be used for later. The 2 pyruvate will undergo oxidation that will produce 2 acetyl-CoA molecules and also production of 2 more NADH. Now the acetyl-CoA go to the next step, which is Krebs cycle. Both pyruvate oxidation and Krebs cycle take place in the mitochondria. In the Krebs cycle, the 2 acetyl-CoA goes separately and produces 3 NADH, 1 FADH2, and 1 ATP. So basically, in each acetyl-CoA, we will have 3 NADH, 1 FADH2, and 2 ATP. Since we have 2 acetyl-CoA, we have a total of 6 NADH, 2 FADH2, and 2 ATP molecules. In the final step, which is electron transport chain, which takes place in mitochondria, this process utilizes the NADH and FADH2. For each NADH that goes to electron transport chain, we will have 2.5 ATP, and for FADH2, we will have 1.5 ATP. If we sum it all, a 1 glucose molecule will have a total of 10 ADH, 2 from glycolysis, 2 from pyruvate oxidation, and 6 from Krebs cycle, which we will have 25 of total ATP, which is 10 multiplied by 2.5, it will result to 25 ATP. Another thing is the 2 FADH from the Krebs cycle, which converts into 3 ATP. Finally, we have 4 ATP molecules, 2 from glycolysis and 2 from Krebs cycle, which results in a total of 32 ATP molecule in a single glucose of cellular respiration.